All right, welcome everybody. Welcome to the Teruko Yata Memorial Lecture. It is our great pleasure to welcome Professor Brad Nelson today. Brad actually graduated from CMU, from the robotics program. There's a couple of notable things about this. He was part of the second class, the second one in 1995. He graduated in 4.5 years, which is a very fast turnaround. Also, another tidbit about his, tell you as a student at CMU, is the only TA I had who actually burned a stack of homework.
That's right.
That's a long story. But he's known for other things as well. He moved on to the University of Minnesota and University of Chicago, and now has been at ETH since 2002, where he's doing fantastic work on nanorobotics with a lot of very interesting results, and I think 5 spin-offs per it. And so Brad is going to tell us about his work today. Thank you.
Okay, thanks a lot, Marcel. It's wonderful to be back here in Pittsburgh, back at Carnegie Mellon. I haven't got back here much. I left 20 years ago, and things change every time I'm here. But it's a wonderful environment, a great place to spend 5 years when I was here. So I'm going to talk today about what's been going on in my group for the last several years. When I was here during my Ph.D., I worked on visual servoing and force control of arms. So I was more, at these scale, meter scale systems. And as soon as I got out, I was trying to look for new directions, new interesting areas to work in. And it turned out in 1995, when I left here, IROS happened to be in Pittsburgh that year. And there were 2 papers that appeared at IROS, one by Toshi Fukuda and Fumihiro Arai from Nagoya University, and the other by Ron Fearing at Berkeley, and both of them were dealing with the mechanics of micromanipulation. What happens when you start thinking about handling small objects, and how does the mechanics work? Because you take manipulation, you look at that, and a lot of times we think of gravity and we think of friction, and all of a sudden we realize as we went smaller, the physics of the world got more interesting, and I was hoping I could find a lot of results from the field of robotics that had been developed over decades and see how they mapped into this micro world. And so I've kind of been there now for 20 years looking at how we can move robotics at those scales. And it's worked out okay. There's been a lot of interesting problems. One of the first things we started thinking about, though, was micro and nano manipulation, understanding the mechanics of manipulation, trying to understand how we could handle things. And after I was in a few years, and then in '98, I saw somebody poking a cell, and I got this thought, wouldn't that be interesting if we could make robots that worked under microscopes handling, in this case, we're doing a pronuclear DNA injection procedure. We're trying to target a nucleus and inject DNA into the nucleus of a cell. And one of the first things I discovered was that there weren't force sensors. I was into force control, and of course we need to sense touch, and there were no force sensors on the market that really gave you the kinds of feedback you needed. And so I went down this road of trying to develop, learning MEMS, trying to develop my own micro force sensors. And still we continue to work in this area and focus and still work with a lot of biologists and try to understand where this can go. But it's been gratifying to be able to take some of this work. And once I got to Switzerland, and then in 2007 we were able to spin off this company called Femto Tools that started with these force sensors, but has gone way off. They have on the order of something like 25 to 30 different products that deal with manipulation, handling, MEMS test equipment, and all these kinds of things. And so after you're in academia as long as I am, you start to hope to see some of your work actually getting into the real world and making an impact. And this is one of the ways, is to try to spin this off into companies. So we continue to work in this area. I'm not going to spend a lot of time on it, but I do think there's some fundamental problems that still exist that need to be solved. One of them is calibration. We all know how important calibration is to robotic systems, but at the micro scale, you don't appreciate what we'd really like to be able to do are take our force sensors and trace them back to that magical kilogram that's sitting somewhere near Paris that represents precisely what a kilogram is, and following this chain and trying to sell a product that can actually claim to be traceable back to this SI standard is a non-trivial task. There have been many, many millions of dollars spent by a number of standards institutes around the world trying to do that. But I think if we can get these products, then that means you don't realize it, but when you're reading biology papers and you're looking at some of these mechanical biology statements that are being made on the forces, they're actually pretty lousy estimates of what's really going on. And if we can really develop products that go back that way, I think it's an important impact that can be made. But I'm not going to go back. You know, calibration is one of those topics which just gets a little, it's not that it's boring, I guess, but it's just not quite so sexy as other things, right? So, but instead what I want to talk about are what happens if we can really make small robots that are tiny and small in scale, and what are some of the things that can be done. We started thinking about this back in, I remember when the idea came that we decided to pursue this. I remember exactly where I was in February in 2003, talking with one of my engineers in my apartment in Zurich, and I had just moved there. And the first ideas you come up with are in targeted therapies, how we can deliver drugs or radioactive seeds or heat or cold or even stem cells now as we see stem cell therapies. How can we get these things to the locations we want? Are there ways we can do this with little tiny robots that are going to be swimming through our bodies doing this? But there's other things we might look at too. How can we put in controllable structures like maybe make stents and microstents, not just in the aorta, but in other parts of the body. Performing occlusions, there's a fetal surgery that sometimes there's occlusions inserted into the airway or the larynx of an infant or tissue scaffolds, or I should say a fetus. Removing materials, ablating it, taking biopsies, or even a telemetry kind of processes where we're searching for maybe the source of blood somewhere, searching for a disease that isn't turning up in your MR or your CT scans. Can we find this with robots or sense, for instance, oxygen at the vitreoretinal interface? So there's a lot of potential applications for this. None of which, of course, we've seen in humans yet, but there's a lot of work going on there. And so I want to tell you a little bit about the path we've taken and where things have gone in this direction. First thing when you think of microrobots, you ask yourself is, okay, how are we going to move things in the body? What's the best way to do it? We'd love for them to have, have their own power source, to be these autonomous beings, but we're not quite there yet, although we're always looking at the literature and trying to find things. But consider one of the interesting stories over the past decade is capsule endoscopy, swallowing these little tiny, they're about 11 millimeters in diameter, about 29 millimeters long, these little tiny camera pills that take a picture of your gastrointestinal tract as it takes its 8-hour journey all the way through, transmits those images. But those pills on the market right now can't be moved. And so this is one of the areas of research people have been considering over the years. What if we could move them? How might we make them move? Well, one thing that you could do is turn them, magnetize them, put some magnetic material on them. And if I brought another magnet external to the body, I might be able to attract it, the opposite poles attract, bring another magnet to the other side, and we can start moving it around. Now, it becomes a little more elegant if instead of with permanent magnets, I can use electromagnets. So I can have, maybe an iron core here, so a soft material that doesn't remember it, and I put a coil around it, winding it, and as I turn on that coil, I put current through it, it turns into an electromagnet, and it sucks it one way. And the other advantage here is I can change the strength that way, and I can also even change the polarity, so I can change the direction. Now the question becomes a very robotic question. How many do I need? Okay. If I want to move this with 5 degrees of freedom or 6 degrees of freedom, how many electromagnets do I want? Well, we all know from robotics that you need, let's see, if I move something with 5 degrees of freedom, I need 5 actuators. Okay. That's what we would think. We'll get back to that later. But in order to do this, we bring in a lot of mathematics and a lot of just classical robotics. And that's one of the things we've been able to do in my group is kind of create this what we consider a breakthrough. But I want to first talk a little bit about magnetic actuation, just some fundamental physics. So if there's two areas of physics I want to make sure you remember. It's how we can generate forces with magnets and how we can generate torques with magnets. And you all know this. intuitively if you don't exactly remember back to your physics where you saw this. And the way we generate forces is with a gradient. That's what the gradient term here is. We're always going, our magnetized body is always heading in the direction of increasing field and increasing gradients. And that's the way we generate a force. Now a torque is like a compass needle and it just aligns with the field. So as we change our field, then our system aligns. And those are the basic physics we need to know. Gradients are giving us the force, and the torque is just being generated by misalignment between the magnetization of my object. And remember, this has to be, of course, something that's either soft or hard magnet. So it could be iron or nickel that are soft, or something like a rare earth alloy like neodymium iron boron that you can permanently magnetize and remembers its magnetic properties. So when we first did this, like all robotics problem, you do it in a plane, okay? So we tried to solve this problem. This is a millimeter diameter chamber. This is a piece of nickel that we micro-assembled using our micro-manipulation technology out of 50 micron thick plates. So these are about a half a hair width thick plates. And we move it through this little maze here and show as we created these generated coils or generated magnetic fields with these coils and gradients with these, we could move it throughout this, and we'd rotate the coils and that kind of thing. And so then the problem, Berk Yeshin did this for us. We actually did this in 2004 and published in one of our most highly cited papers. And then I brought in a postdoc from Alison Okamura's group by the name of Jake Abbott, and his job was, I says, okay, take this 2D problem and make it 3D. So we all know that the extension to 3D is trivial in robotics, right? And it turned out it wasn't, of course. But what we did is going back to these equations, there's two important things I want to remind you about. There are two primary terms here in these equations, and one is the volume of the material and its magnetization. Now this magnetization, M, is a function of the material you're using, its chemistry, the elements it's made of. It's also a function of its shape. So that brings some interesting design opportunities here, whether you've got an ellipsoid or a sphere or some other strange shape. So that's one of the important terms. And I've got a whole chemistry group, a materials and fabrication group run by a physical chemist, an electroplating specialist, and their job is to deal with these. That is, give me the right properties, the right magnetic properties, and give me the shapes, the structures that we need. And that's a whole area of research, and it's very fruitful for us. I'm not going to spend a lot of time on it, talking about it, but that's a big part of this equation. Now the systems part comes into generating the fields and the field gradients and how we represent those. And that's where more of what I consider more of the robotics side comes in. And one of the things that Jake was able to do, along with Michael Klummer, one of my students, was cast this problem in a fashion that looks very much like a traditional manipulator Jacobian, where this I've got now n instead of joint angles or velocities here, what I've got are n currents in my n electromagnets. In this case, I'm showing 3, but I can have an arbitrary number in this vector. Maps through this mapping, which consists of the magnetization of this, and this is again, coming back, this is a very nonlinear function in fact, but there are ways we can linearize or consider it constant at certain certain circumstances. Also the position of the device in the workspace, and I can then generate my torques and forces. So you see what happens is when we were able to do this, publish this in 2010, we were able to go back to where back in 1985, if you go back that far, you can find the papers from people like Tsuneo Yoshikawa at Kyoto University who showed us how to handle redundant manipulators. We can use work that goes back 30 or more years now, bring all that robotics result, and start thinking about this problem from that perspective. We've got non-zero null spaces in here. We have redundancies, and so we can start thinking about how to use those in ways that really the whole robotics field is brought in. Nobody's been able to do that before. And this is something that we were able to patent, and ETH owns a patent on that. So the very first system we built in 2009, this is our prototype, we call it the OctoMag because it has 8 electromagnets. Here's 4 and here's 4 underneath. You'll notice it's in a half sphere, it's a half plane. And basically what it's able to do, I'll explain this a little bit, is it's able to generate arbitrary fields and field gradients within a workspace. And so what you'll see are these little vectors. These little vectors represent the direction of the magnetic field, and then the colors represent how the gradient. And these vectors are actually, it's hard to see here, but they're actually getting bigger as you move towards red, and they're getting smaller as you move towards blue. So there's a lot of math behind this. These diagrams here, these little bars are showing the currents. They're a representation of the currents in the 8 electromagnets, red being one direction, blue being the other, and of course the length is the magnitude of those. And so we were the first to be able to show how you can arbitrarily move something in 5 degrees of speed, with 5 degrees of freedom in space. And so, like I said, that was something patented. Now, we were kind of lucky to arrive at 8. We did this through a lot of numerical simulations, and we realized if we started with 6, we had a lot of issues with control authority, these kinds of things. We didn't quite understand why, but we realized when we moved to 8, all of a sudden we got a good, much more controllable type of a workspace. So it wasn't until just this year that we finally understood this analytically. And I want to go over the math here. This is a paper that will be coming out in TRO soon. And the first author is Andrew Petrushka. Now, it turns out Andrew is one of the Ph.D. students of Jake, who was in my group. Now, Andrew also got his bachelor's in physics and engineering from CMU and did his master's degree here as well. So he's a CMU grad. He's working for me as a postdoc now. The thing I want to point out is, in this figure is I've published, I've been around a while, so I published lots and lots of papers, over 300 papers. This is the first paper I've ever published that doesn't have a single figure in it, okay? And that's why I show this, okay? I'm very proud of that for some reason. So we can move things, okay? So what are we going to do? Where are we going to do this in the body? Well, one of the first people to approach me and talk to me about ophthalmic surgery was Cam Rivera. Also worked, with Russ Taylor, Greg Hager, some of the folks at Hopkins. And so we started thinking about what might we do. Could we do anything with this system in the eye? And so this is a video that came out of the Discovery Channel. Discovery Channel made several years ago for us. I still use it because it captures the idea very, very succinctly. Here's our OctoMag above the patient. And now this is looking into a pig's eye in this case. This is about a 0.5 millimeter in diameter. This little permanent magnet, about 1 millimeter or so long, and just moving it through, you're looking through an ophthalmic microscope here. And so this kind of gives you the concept of what we might do. We might be able to inject things into the eye of a patient and move them around and do something with them, okay? And I'll tell you a little bit about what we might want to do. One of the things we've done over the years, several animal trials. This is our small animal system where we have our OctoMag now. It's at an angle. We bring in the animal, in this case, a rabbit whose eyes are about half the size of a human. Maybe a little bigger. Here's an ophthalmic microscope with a special lens that will look through there. One of the design criteria for our device is that as we inject this into the eye, we want it to fit into a needle that's no bigger than 23 gauges. And so that limits the diameter to about 300 microns, so about 1/3 of a millimeter diameter. If we can make it that small, then these injections can be sutureless. So as you inject it in and you pull it out, the wound closes itself and we don't have to do injection. We'd love to be able to go even smaller, down to 27 gauge or something like that. But at this case, remember that V term in my physics equations, volume. Volume's a big deal here. If we get too small, the forces just get too small. So this is one of our devices. This is in a model eye in this case, and you can see looking down, moving through the ocular cavity, all the way down to the retina in this case, and moving along one of the major retinal arteries or veins here. So to me, it's a bit like a spaceship moving and landing on Mars or something like that. And a lot of the way you use this, a lot of the issues in user interfaces and how do you present this to a potential surgeon that might be using this can be done. Also automation, but you'll notice one thing is there's visual servoing involved here. Okay, and if you think back, that's what my Ph.D. was on. A lot of what I do still has visual servoing in it, but I almost never mention it still, but we still do a lot of that. Now, one of the things as you go to real tissue, we go to real animals, you realize is that it's one thing when you're moving through oil or water, which is very homogeneous and very Newtonian, But in fact, real tissue is often non-Newtonian and often has some very nonlinear properties. And one of the things about the vitreous humor, this jelly stuff in your eye, is that it's got actually two types of fiber networks or polymer networks in it. One is this tiny little network of hyaluronic acid, and the other is the larger network of collagen fibers. And so one of the big headaches in our group has been trying to deal with and understand the nature of that vitreous humor, because what happens as you're moving through it, instead of moving so nicely as you saw in those others, is that it gets wrapped up in some of these fibers and creates this viscoelastic nonlinear approach. And so a lot of our effort has been in trying to understand the biomechanics of this tissue, and that's been a lot of the experiments that have gone on in our group. We do that with animals, and then we've also do it with human donor eyes. And so this is some examples. We've gotten a couple of donor eyes. In this case, these are examples from the hospital in Bern in Switzerland. And one of the interesting things about the vitreous humor is that it changes with age. So if you're very young, it's very elastic and rubbery. And as you get older, it gets much more watery. And so we can verify that as we use donor eyes. We can then create microreology data and try to match that to biomechanical models and verify that that's true. So now at least we know something about our environment and how to control things as environmental parameters change. And so again, not just controlling this device in space with the OctoMag, but also understanding the environment it has to pursue or has to move through is also important. Here's an example of, this is actually done this is a chicken embryo vein showing a venal puncture. This is one of the things we like to do, is inject drugs directly into the vein. And these vessels are about 125 microns in diameter here. They're about a little bit bigger than a hair width. So you'll notice our device being able to float and then puncture that very precisely exactly where we want. And you can imagine, this is one of the things that's so phenomenal about surgeons' capabilities, is that they can keep their hands steady enough. If you went in and tried to do this, it's extremely difficult to do by holding the probes. Surgeons can do this, but we can go even more precisely than some surgeons have been able to demonstrate here. Here are some results of some of the animal trials with rabbits. This is one of the first devices we put in. Rabbit eyes, it turns out, I didn't appreciate this till we first did it, have huge lenses, so it's going to look very magnified because of the nature of the lens. But this device is about 1/3 of a millimeter in diameter, about 1.8 millimeters long. And here it is actually moving in the real vitreous humor of a rabbit. So not all rabbits it would work in, but some of our subjects we were able to demonstrate moving in intact vitreous. And that's important because if you ever have retinal surgery, if you've ever gone to a surgeon to do this, the first thing they'll do is they'll suck out your vitreous and replace it with usually a saline solution. Now that can have some complications of its own. It also takes a while to do that. So if we can develop devices that don't require that, then we've given an advantage to the procedure itself. So these are some of the things that, some of the procedures, advantages we think that this could bring to procedures in the future. So I'm not going to belabor with ophthalmic microrobots, but I want to kind of catch you up to where we are in this right now. What we've developed so far is a microrobot that can navigate throughout the human eye. In fact, right now I'm building a human-sized system in my lab. It should be in operation in a month or two, so you can fit an entire human into it. We've demonstrated micron positioning precision. No one yet, and this is changing very rapidly, no one yet has really demonstrated force feedback, and it's not used in surgeries yet. With our system, we have inherent force feedback in it because we know exactly how much current we're putting into our coils, and that maps very directly into the force that's being generated by the device. Unlike where you have to go through a kinematic chain and come back. So we've got inherent force feedback there. The injections require no sutures. So that's the way surgery is done today with these. Potentially no vitrectomy. So we've demonstrated the ability not to have to suck out this vitreous. Drug delivery, we're looking at one of our first targeted therapies is delivering drugs to the retina. We've got a couple different types of very specific therapies we're looking at and working with surgeons on. And finally, we think there are a lot of other possibilities, and we're pursuing, for instance, peeling little membranes off the back of the retina, which is a very common surgery that needs to be done, repairing retinal tears, precisely placing retinal implants. And we're also looking at the anterior segment, that is the front part of the eye too, and using some of these in these. in the front part for types of cataract surgery, or looking at underneath the eye where actually the source of glaucoma is in the Schlemm's canal there. So this is kind of where we are at this point. There's other projects going on in this area, but I want to move on and talk about some of the other efforts that have been going on. And one of the other locations we started looking at was the heart. Now, one of the problems with the heart is that it's beating, and it's got a tremendous blood flow through it, right? The blood coming out of your aorta is on the order of 70 centimeters a second. So there's, you're not going to be able to put little devices in there and hold them against the flow. But one thing we realized, though, was that this magnetic manipulation procedure that we've been able to develop and patent gives us an ability now to, instead of, if we take those robots and we put them on the end of a catheter now, we can steer those catheters very precisely. And so this is a setup I have in my lab back in Zurich in the basement. This is a 6.5-ton electromagnet. It's got 8 electromagnetic coils. They're about this big, and they each are about 500 kilograms each. It's big enough to hold a small person or a 70-kilogram pig. One of the things you'll notice is this is all X-ray protection, because this is all done under fluoroscopy, under X-rays, a lot of these procedures. And so what we've been able to demonstrate is, I'll show you a fluoroscopic image here in a bit, is that we can take these tiny catheters now and we can generate very, very high radius and very highly dexterous motion by exerting forces and torques on the tip of the catheter. So we've basically taken our micro-robot, that right here, and put a tether on it so that we can then put it in and out and move it throughout the body. So this next little video I'm going to show you is where we are with this project right now. That we've spun off a company called Aeon Scientific that's licensed our patent and some of the other patents we've developed in this area. And they're looking at a procedure called cardiac ablation. So I want to show you that in this little video. I think there's sound here. This is a system that's actually installed in a hospital right now, and sometime between May and July we'll start using it in humans. So. Is there a volume here? Oh.
The modular design of the Aeon focus allows for the flexible use of the
Hope you can hear that.
The physician can operate the system remotely from a control room, where he is safely protected from X-ray radiation. With the two halves of the system parked away from the patient, the physician has good access to the patient and can make full use of the angiography system so that he can comfortably insert the required catheters through the sheath into the relevant heart chamber. After completing the catheterization and of the X-ray fluoroscope. Now the electromagnets can be activated to generate non-harmful magnetic fields. The magnetic fields can be set to any direction within the patient's torso without needing to move the electromagnets. Using the resulting magnetic forces, the physician can steer the tip of the ablation catheter with high dexterity and unprecedented reach within any of the 4 heart chambers. Such control over the steering of the catheter is key to successful ablation. With the intuitive control and the dexterity that the Aeon focus provides, the physician can quickly target the desired positions within the cardiac chamber. Thanks to the precise and stable contact, continuous and transmural lesions can be ablated, leading to a successful outcome. The Aeon focus provides a new level of control in catheter steering during mapping and ablation procedures. With its open and flexible design, the system adapts to any workflow in the cardiac electrophysiology laboratory, and it will bring considerable benefits for patients and physicians alike.
Okay, so that's a system that's obviously an animation, but that a system looks very much like that, is now in a Zurich area hospital, and should be first use in humans sometime in the next few months. I didn't go into detail, but it was treating a condition called atrial fibrillation, and one of the reasons that was selected by the business side of this was that these are just increasing cases, and they see a possibility for commercial feedback to develop the system in that case. But we see this as a kind of platform technology for many other types of catheterizations, and we're looking at endoscopic procedures, looking at things, for instance, in intravascular neurosurgery or in gastroenterology, in bronchoscopy. So I think there's a lot of other things that can be done here, but this is the first very focused application that we're looking at there. And we're hoping then, with the success of this, to continue to grow this. And so, as a company that's spun out in 2010, and that's where they are right now. But let me, yes, sir. Yes.
So there was a company called Stereotaxis.
Yep.
And why did they not do well, and how is yours different?
Okay. Yeah, Stereotaxis is out of St. Louis, Missouri. They've been on the market for over 10 years. They have, for some reason, I want to say 103 systems out there, or something on that order. They use big permanent magnets. They have 2 permanent magnets on each side of the patient, and they move. And I can give you more detail about that system and why it's, let's see, they've quit posting their sales figures now. But you can look at their stock price if you want. Yeah, there's some issues, and I think we have to overcome. There's another competitor out there too called Magnetex, and so we're also considering them. But this area of ablation procedures, this is I mean, people see a potential for a large business in it, and we haven't quite got the right technical solution. We hope that maybe this will address that. So.
Also, so in the video, it said that you were able to get transmural ablations.
Yes.
So how do you know you're getting transmural ablations?
So we did either 15 or 18 pigs, and then we sacrifice the pigs afterwards, and we look at their hearts. But also, you can tell as you're looking at the electric signals coming out of the heart. You can tell when you're the physicians look at that, and they can, or the electrophysiologists can look at that, and they can see as they're ablating if they're getting that. But one of the points of the animal trials was to look at the lines, the paths they were taking to make sure they were transmural. So.
Right. So there's nothing in the device itself that there's something you feel.
There's two catheters on the market that will sense forces, and not sold by Aon, but sold by other companies. One of them, yeah. And those are, I think one's piezo-resistive, and the other uses a fiber optic sensing. And I think that's new. I was just talking to a big electrophysiology group out in the other side of the state a while ago, and they, I'm told all the new EP guys who use these don't go back. They really like the force feedback and seeing that they're getting enough force. So, okay. So I want to kind of finish up here and talk about what we're doing in going smaller. How can we make even, so we started with the eye with millimeter, sub-millimeter sized devices. We scaled that up to larger things, guiding catheters, but we're also, a lot of more of the fundamental work we're doing is going smaller and trying to understand, can we make robots the size of red blood cells, and can they do anything interesting as well? So it's one of these areas that, from a fundamental standpoint, is very rich, and we're trying to make that move beyond that. What we'd like to do is make maybe go even smaller than red blood cells. Make robots the size of bacteria. These are maybe a micron or 2 long, very, very tiny devices. Can we make devices kind of model them after bacteria? These bacteria that move and swim. Now, nature's, of course, created a lot of microorganisms that are very small and are able to locomote through their environment. For instance, paramecia. These guys are around 100, 150 microns diameter. They have on them lots of these little cilia, hundreds, maybe thousands of these cilia that beat in this kind of synchronous fashion to propel itself through this liquid. And you look at this guy, and he's searching for food, and he's avoiding obstacles. He looks pretty intelligent. There's not, of course, it's a single cell. There's not even a neuron in there, right? So what's going on? This is spermatozoa. Again, very small, a micron or 2 in size. And you look at this tail that's whipping, this eukaryotic flagella that's whipping. There's a very specific mathematical reason it's moving. It's got a traveling wave, and there's very good reasons given the low Reynolds number, the type of fluid dynamics it has to overcome here. So these are 2 examples of microorganisms that swim this way. But one microorganism particularly interests us is the bacteria. For instance, bacteria like E. coli or Salmonella that can flagellate and become motile. And they're on the order of a micron or two long. Now, these were first seen under a microscope by Anton von Leeuwenhoek in 1675 or something like that, well over 300 years ago. And whenever anybody looked at them under a microscope, you could see their flagella moving, but it would look like they were moving like a traveling wave. But it wasn't until in the 1970s when Howard Berg was at University of Colorado that he realized they were not wiggling back and forth, but they were actually rotating. And so what Berg had discovered was a rotary motor. And this was a phenomenal discovery at the time because nobody could imagine how could nature, what in nature has a rotary motor? Nothing people could come up with. You look at large-scale animals, nothing rotates like that. And so that was a breakthrough. So we looked at that organism, and as you study the motor, it's a fascinating structure. Ever since Berg discovered it, molecular biologists and geneticists have tried to understand what it's made of, and they know all the parts. There's still some questions on the way itself assembles into this device, but it's made of about 30 to 40 different kinds of proteins that come together, and it's driven by protons. It's driven by hydrogen ions charges that move through it. And here's a fluorescent video with the head and these flagella, and you see them swimming in this case. So those are pretty interesting. Now, from a nanotechnology standpoint, this guy's about 45 nanometers in diameter. I can't make anything quite like this motor. But we started looking at the flagella, and we started looking at some of our other nanotechnology efforts. And we had become interested before in looking at strained semiconductor layers and getting them to form into interesting shapes, to kind of self-assemble. So we would take some type of semiconductor, in this case silicon and silicon germanium, put a little chromium on top to give it some stability. And if we got the crystalline planes lined up, and so there is also a mismatch in the stresses here, we could get them to actually form these interesting structures. And one of the structures looked surprisingly similar in size and shape to these bacterial flagella. And so we started looking and thinking, I wonder if there's a way we could use these as flagella to propel small devices and so while I said I couldn't make the motor, what we could do and what we knew something about was magnetic materials. So we took our tiny little flagella, our tiny little springs, and we put a little piece of magnet on the end, a little nickel on the tip of it. And if you go back to those physics equations, you'll remember about the torque. And if I generate a torque by taking a field and rotating it, so I take my magnetic field, forget about gradients now, and I'm just rotating that field, what happens is I can generate a propulsive force. So it starts moving like a propeller. There's a lot of fluid dynamics that's fascinating at these scales. If you're a mechanical engineer or physicist, you're familiar with Reynolds numbers. Jets, 747 has Reynolds numbers in the billions or so. These Reynolds numbers are like 10 to the minus 4, 10 to the minus 5, very, very low. And that has an important impact on the way these things move and the physics of them. One of the cool things about this, though, is we found out these fields that we generated were very, very low. They're on the order of 1 to 10 milliteslas, so they are about 100 to 1,000 times less than MRI type fields. And we could rotate them. So we can project them through your body and make these things rotate. So this is a micrograph of a human hair. These are the smallest flagella, we call them artificial bacteria flagella, or ABF, that we've been able to make. And this is a red blood cell. So we're getting to the dimensions of red blood cells where we can make things that small. And here are some of the first devices we made. This is many years ago now, 2000, probably this is videos from 2008 or so, 2009. You'll see them, these guys are about 50, 60 microns long, so they're about as long as half the width of a hair. Now, one of the things you'll notice is they start moving backwards. What we did was we just reversed the rotation of the field, and they go backwards. And you can see, you maybe get a sense of the fluid dynamics here. You can think of this, imagine yourself swimming in a vat of honey, okay, or something like that. That's what water feels like to these because they have essentially no inertia. They're just feeling these really strong drag forces from their environment. And that has a huge impact on their design and their optimization. And so we spent a lot of time trying to understand the physics of why this is going on, optimize fabrication procedures and these kinds of things. Now, as we had, remember, these were made from semiconductor layers. These are made from things like indium gallium arsenide, things that are toxic and you don't want to put in your body. Oh, I should mention, we also, in the Guinness Book of World Records in 2012 for these guys, which is great. Here's some of our other ones there where we're trying to build a synchronized swimmer. This is real time, by the way. So as we go down this path, and we first used semiconductor equipment processes to make this, but after we'd started developing these, then we discovered what I call 3D nanoprinting came on the market, and we started to use 3D nanoprinters. So I'm going to let this run a little bit because it's got a really dramatic finish to it. But they look like they're alive. They look like they're alive to me. I don't know. So anyway, a lot of shameless advertising in this talk. So anyway, one of the things that happened as we're going down this path, though, is new technologies come along. And one of the things that came on the market was this 2-photon photopolymerization project. Basically, you take a photosensitive polymer, and by basically with a laser, you could just write whatever shape you want in it. But you can do this with micron precision, even sub-micron precision. This is started being sold by a company called Nanoscribe out of Germany. We were lucky to get one of their very first machines about 5 or 6 years ago. And that opened up tremendous possibilities. So instead of being limited to all these microfabrication flat processes, now we could actually develop and design 3D devices and 3D print them essentially. We also could use polymers. And so we could start thinking about biocompatibility and start looking, and we can also make them much cheaper and easier. And all of a sudden our process, it opened up a vast array of capabilities for us. And so basically the idea is, as I mentioned, you start with this photoresist, you write in it, and this is real-time writing with the laser in it. It takes about a second or two to write them. We bake it a little bit, wash off the photoresist, and then we coat them with nickel. That's the magnetic part. And then we coat that nickel with a thin layer of titanium to protect it from oxidation and to give it a little more chemical stability. And also it's biocompatible. Titanium, cells will, or at least it's non-cytotoxic, cells can grow on it. So this gives us a lot of interesting capabilities here that we can make. One of the other things we did was we took our OctoMag setup, remember the big thing that we have patented, and we made it small enough to fit under a microscope. So we've got our 8 electromagnets here, and we can generate arbitrary fields and even field gradients and start looking at that. And so this is another example of a device then we've been able to turn into a company and sell these. And so this just came on the market last year, and we're just shipping a few products right now and getting a sense of what possibilities there are for this. One of the things we're looking at is X-ray crystallography and handling delicate protein crystals with that. So now what happened was we started off with this procedure which required very complicated microfabrication techniques like molecular beam epitaxy and also specialized coils. And now we've got is an ability to make, with a Nanoscribe, 3D print whatever we want. And with our Magnobotics system, we're able to then very precisely control these in very nice environmental conditions. So now that's opened up the ability to do a lot of other types of experiments. One thing I want to mention about the physics of this is, and the fluid dynamics of these swimmers is basically as you rotate these with the speed, you get a forward propulsive motion. We talked about that earlier. And it's a very nice linear kind of representations, and we can do this in 6D, but I'm just showing you the planar capability here. But we understand these parameters in this matrix are a function of the viscosity of the material you're in, assuming it's a Newtonian, of course, at this point, and the geometry of the device. And as you'll notice, if we look at this plot here, the faster it rotates, the faster it goes forward. You would expect that. But there's two design criteria we want to look at here in optimizing these. One is the effective pitch. So this is like a screw. You turn it and it goes forward one turn forward, a pitch forward. But there is always going to be slip in the liquid. So you're going to get somewhat less than the real pitch. So you'll get an effective pitch that's and you want to optimize that. We can determine that from here. There's also a step-out frequency. The faster you rotate these, the higher the rotational drag is. And at some point, you can't overcome the rotational drag from the magnetic force. And so we get like a step-out frequency like with a stepper motor. If you rotate a stepper motor too fast, you have too high of a load, it will stop, it'll step out. So we get the same kind of effect here. So what we want to do is optimize our omega max here and then get as close to 1 as we can with our proportional efficiency. So that's one of the metrics we have of performance for these devices. The other thing we spent a lot of time on, so we can design things very precisely with that, the other thing we spent a lot of time on is functionalizing them. Now we can make them swim, we understand something about the fluid dynamics, and what are we going to do with them? So we've taken, in this case, we've taken one of our ABFs. These are these are actually mouse cells that are stained red. They're not red blood, they're mouse cells. And we can put a fluorescent protein, in this case it's calcine inside of it, and track it as it moves through. The other thing we can do is we can make armies of these, because as I showed you writing, we're writing them, you know, one or every second or two we can write one. And so if we turn the machine on and run it all day long, I can make 50,000 in a day. So I can also make swarms of these. So we're starting to get a handle now on how to functionalize, how to attach molecules to them. Also how to get these things to come into contact with cells and get the cells to take in the drug or the molecule that is being held on here. And so that's where we are right now. We've taken this paper just has come out recently where we've taken what we call these now functionalized ABFs. We coat these with a liposome complex, which basically it's like a cell membrane itself, and in that we can also put our drug molecule. And so then when these come in contact with cells, in this case we have fluorescein on this particular one, a fluorescent protein. As we come in contact with all these cells, you'll see one of my ABFs right here sitting on these two cells. These cells take in that this particular DNA, take it into the nucleus and can then express it. And so that's showing gene transfection. So we're able to actually load DNA or RNA onto these devices, bring them in contact with the cell, get the cell to take that up, and then start expressing that particular protein. In this case, a Venus protein that has DNA in it. So this is something which recently just appeared on the cover of Advanced Functional Materials, and this is a confocal microscope image. These red marks are the coils of the ABF that's sitting on it, and then you'll see these fluorescing, and the surrounding cells then are not expressing that. So this is something we're able to demonstrate gene transfection. And then finally, we showed it with one, but can we get lots and lots of these? Can we inject many into a living organism and get them to move? And so in this case, we took our ABFs and we attached a near-infrared dye to it. And we injected, in this case, 80,000. So this is a near-infrared dye, which if you fluoresce it, then it will actually penetrate the skin a small depth. And so we put it into a mouse peritoneal cavity. This is the whole mouse, and then this is with an IR camera, seeing this swarm in there. And so what we're able to do now is localize these. So now think back to visual servoing. Now I have a way of putting a bunch of devices into an animal or a human, and I can actually observe them with computer vision, okay, and I can control them and track them. And so that's what you're seeing here is a swarm of these, again 80,000. This is actually done, this is an example of seeing them in vitro. We can't pick out individual ones inside, but we can pick out clouds of them inside and move, in this case, like I said, 80,000 of these at microns, a few microns per second throughout in the peritoneal cavity of a mouse. So now what we've been able to demonstrate is gene transfection with these, as well as basically visual servoing clouds of these within living organisms. And so this is something that is just coming out right now. It'll be on the cover of Advanced Materials, one of the high-impact materials journals sometime. I'm not sure when it's coming out. It's all been accepted and camera-ready copy and everything in. So look for that soon. Okay, so I want to just summarize, kind of tell you where I think we are and what our accomplishments have so far been in the field, and not just us, I mean the whole microrobotics community. I think the primary application area that we've been looking at so far has been in medicine, and we worked a lot with biologists. We also are working on some manufacturing application And lately I've been thinking about environmental monitoring, or even water remediation, where we can inject, we can put thousands, millions of these into maybe wastewater or polluted water and catalyze pollutants somehow. So we're starting to move in those directions with it and see some other potential applications, hopefully, down the line there. You know, I think it's a field that's still pretty new. I just started thinking micro and nano when I left here 20 years ago. We really didn't see people start thinking about real autonomous devices, though, until the last 10 to 12 years. And we've been able to now to solve, or at least understand how to address some of the issues of powering these devices, how to get them to move, how to fabricate them. And also looking at appropriate therapies. We talk to doctors, we work with surgeons. Every time they see this stuff, there's a lot of ideas. And so we're starting to head down some of these paths to try to get these out of the lab and into the real world in some sense. I think it's a field with a lot of potential still. There's still a lot of, too many problems to work on. It's still a lot of fun. It's got an uncertain timeline, though, because you never know how the breakthroughs are coming. And if we're really gonna really make an impact in the clinics, you know, it's not just about the science and technology. You've got to get the doctors on board first. You've got to see business cases, because it takes tremendous investment. And so you got to think about business plans, and you got to think about does it make sense to try to spin off a company and attract investors to it, and how is this all going to come together? You also got to deal with regulatory issues too, of course. But I think it's a field in its infancy. Still got a lot left to go. And we see more and more people entering it, and more interesting things happening every month, or certainly every few months, something new is coming out. So that's what I wanted to tell you about. I've got a very great group. I got about 30, 35 people back in Zurich. This is Zurich, by the way, Lake Zurich. These are the Glarnerland mountains. Some of these folks actually have been through here at CMU. Some of these folks have come from CMU. One of the things that's been nice at ETH is that I've been able to bring together a very diverse group. So while we're, you know, first roboticists, we also have biomedical engineers. We have microfabrication people, chemists and physicists, a lot of nanotechnology people. The problem when I spin off groups is my computer science side tends to, my software people get decimated. They all get sucked into the spin-offs often, and I always have to rebuild that side of it, but eventually we get back to there. So there's a lot of things that have been able to come together, and that's given us a kind of interesting perspective on how we can go down this field. So thank you very much for your attention. I'd be happy to answer any questions.
So this, you could argue, is a very engineering approach. I'm going to power some matter somewhere.
Yeah.
On the biological side, the dream has been, let's use monoclonal antibodies, let's throw things that will go everywhere, and they essentially, the ones that are in the right place do the right thing, the others, you know, eventually exit the system. So would it be better to have some synergy between the engineering and the biology?
Yeah, well, we're getting some synergy as we're, you know, and that's happening. I mean, I just, in the last year, we started a cell culturing facility. I certainly keep my eye on the synthetic biologists. I have for the last 15 years. Richard Murray just visited, and, you know, he's been working in this area, and, you know, I still think, I mean, you know, if you look at MIT, there's a ton, you know, they're going that way whole hog, I think. There's a lot of synthetic people out there. But I think, I don't think the engineering, you know, I don't think it's one or the other. I think, as you said, you got to bring them together. The other thing about this that I think sometimes people lose track of is, you know, it's cool to see these things swim, and you think, oh, I'm going to inject them into me, and they're going to go where we want to go. Well, it doesn't work that way. They don't swim very many body lengths a second. So you get them close, and then they have to reposition. So I think there's a lot of issues that need to be looked at, but you certainly want to bring these two together. You know, and I'm always excited to see what comes out of synthetic biology and some of these efforts, but I still don't think the case is closed on it. So.
You spoke a lot about and showed a lot of injecting these things and moving them around, but you talked about how you would remove them, starting with the larger ones, and then you have these super tiny ones. Do you remove them all?
So with the big one in the eye, we have a little tool that has a little magnet. You put it in like a little syringe, just like they do for injections, that you bring them back and sticks and you just suck it out. So that one you can do. One of the things I haven't talked about, well, we've experimented with, we use a polymer called Ormacomp, which is FDA approved, with iron. So these could stay in the body theoretically for a while, or forever. There are new materials being developed, new bioerodible materials, and so the idea for the large ones obviously are they're just going to erode eventually. You're going to make them put iron for magnetization, use some type of bioerodible. I also work a lot with hydrogels, and I haven't talked about any of the hydrogel work, but a lot of those are bioerodible. So yeah, you either want to, if they're larger, put them in, take them out, or let them erode. And so these are materials issues, right? And so that's why we've got a whole group, about 8 to 10 people that are just working on those things. So.
I have a question about tuning the swarm properties, or have you guys looked at picking out individual robots in the swarm or changing the way it's distributed because you have only one magnetic field?
Yeah, this is that problem of, many robots, one command, right? And so there's some interesting work in the robotics community there. One of the people we talk to a lot is Tim Brettel at Illinois, who's done some interesting work. If I have, n robots, each with different initial conditions, and I can give them one command, how can I get them into a final configuration? That's interesting. One of the other things we've done is, if we change the sizes of ABFs, the drag does not change similar to the magnetic properties. So at certain sizes, at certain frequencies, some will stop moving because they can't overcome the rotational drag from the magnetic torque, whereas others will keep moving. So I can create classes of devices, and I can get some to stop and some to not. And I've seen some work. There's some unpublished work out of Max Planck where they're doing some sort of it's almost like a sailboat tack. Anyway, I'm going to go in. But yeah, I think those are interesting issues, and we think about them. Ideally, what I really want, I want each of my devices to be autonomous, okay? And I got a paper that's about ready to come out that at least demonstrates a step there. So we're using a different kind of material that starts to change based on its environment, and so its drag can change and properties can change, and so we're just finally it's an idea that we've had for about 6 or 7 years, and we're finally just getting the materials, chemistry right to be able to get something out on that. So.
How much of the building blocks of life in robotics sperm really have? Do you need sperm from a human that's going in? How much can you make? And then if you have that, can you replace the DNA in it? It's just going to be like, I'm pretty sure that's one there. And be able to replace the DNA and it's going to
well, I think maybe we were talking about it. Maybe it's more on the synthetic biology. This is what the synthetic biology folks are trying to do. I mean, I'd love to take, you see that rotary motor I showed you that Howard Berg discovered. I'd love to take that chunk of DNA from be able to make just those parts and get them to assemble and do these things. Now, of course, the synthetic biologists are getting better at I mean, they're very good now with CRISPR and some of these techniques for cutting and getting just exactly the parts they want. So I think, yeah, that's not what we do, but there's other people out there that are doing these kinds of things.
So you mentioned a couple of spin-off companies from your technology. Congratulations on that. So I'm curious, what are those companies doing now? I would imagine there's something between leaving your lab and
Yeah, well, I wouldn't congratulate somebody on spinning off a company. I would congratulate them on selling it at a profit or something like that. But spinning off a company is always exciting. But what the companies are doing now. So Aon is, like I said, they're just starting their first use in humans and designing clinical trials. And so the clinical trials should start this fall for their device. And of course, they are in investment round. They're always looking for money, right? There's always the next investment round they're looking for, and it's not quite the same system in Europe or Switzerland as it is here. They started off with an angel, and so they've had angel funding and some small investors along the way, and now we're looking to ramp up and making the case there. FemtoTools is one of the ones. They started off very Swiss-like, very, very conservative and slow, and now they finally converted the last couple of years and are trying to ramp up quickly and just got a major product out now. And So to give you an idea how big these companies are, Aeon has about 19 employees. Femtotools is probably 9 or 10. I showed MagnaBotics. That's just by the string of our teeth right now. We're just getting these devices. One of the advantages we've got, and I'm not sure what it's like here exactly because I've never been on this side of it, but one of the things that's nice about Switzerland and Germany is these engineering firms you can go to with a chunk of money and you say, I need this design and manufacture, and I need a CE mark on it. And so you can bring them devices and they'll work with you on patents and things. So it's a very interesting the way you can do things there. Maybe a little differently than here, I'm not sure, but so you do that, yeah. I was just in the UK with at Guangzhou Yang's lab, and he's like, we don't have those things. We have to in London. He says we have to go out and do that. But.
So this one, the question was, you just use this sterile catheter, it's Aeon.
Mm-hmm.
Did you do additional, what kind of additional development did you have to do after that mechanism left your lab, the company, to make it suitable for a person treating?
So one of the things about, I don't know how it works here, but one of the things at ETH they allow companies to spend the first 3 years in your lab. They rent space from the university and they work. So we kind of get them off the ground, then they go out. And so then basically what they did was they did all the quality assurance, the quality management, go through the whole, all the paperwork to get the marks and the medical device directive stuff. They do all the stuff that's very, very expensive that you can't publish, right? And so that's what they're doing, and then they're working directly on the sales with the hospitals. And then what I do is I've still got my prototype device we use for animal studies in the basement, and I'm looking at other applications for it. And I'm trying to find out, does it make sense to do bronchoscopy with this device and how? So, that's kind of the strategy right now. They are the ones that overcome all the really hard industry hurdles. Our job is to, play with it and see what we can do and investigate things and we get some patents. It's hard, let me tell you. It's a lot harder than publishing a paper. So,
so male and then behind that.
So about this problem of multiple devices that you want to move independently, seems to me there are more degrees of freedom than you know about, or at least more degrees of freedom than you mentioned. Because a real magnet is not a simple dipole. It has quadrupole and octopole and decapole moments that interact with gradients and gradients and gradients. And you could therefore tailor the vehicles with different mixes of these higher moments and have different handles on them. Have you thought about that?
We haven't gone that route, no. One thing we have done is we created resonating structures. And so some of them have resonant frequencies, at 2 kilohertz, some at 5 kilohertz, and so then we can get them to, and these were the ones on the surface. That's actually this guy right here, this Pac-Man. This guy's about 70 microns on a side, and he's resonating as he's moving, and then I can create different ones. These things all in theory are great. In practice, of course, to get the good high Q, you want that nice, is always a lot more difficult than we think. But
also the magnetic field induces another dipole.
Yeah.
Big issue, especially if you're in blood, which has interesting magnetic properties itself.
Well, and a lot of this stuff there's a lot of work in magnetics, right? And we got lucky the first time we got our swimmers to work. We didn't realize, and then we go back to a paper. You take a plate, and if you magnetize it, if you put it in a magnetic field, its easy axis will be along the diagonal, unless it's too high of a field, and then it's here. Well, we didn't know that. We were just lucky enough not to use too high of a field, and then we go back and we find the paper in the 60s that did all the math and showed why.
Yeah.
That's a lot of fun actually when you realize that. But we also realize that one of our sayings is we'd rather be lucky than good, and we get lucky every once in a while, right? So.
Yeah, go ahead.
So, have you taken any steps or even thought about going forward how to actually use active manipulation on one of these devices? Do you foresee any way to do that with magnetic fields, or is that going to be something completely separate, some kind of a chemical trigger, atomic trigger? If I want to have a chamber that I can say, now open that and let the chemical out, how do we do that?
Okay, so I've got two answers to that. One is we've created these kind of claw-like structures out of hydrogels. They're two layers, and hydrogel is something that will swell. You can tune the chemistry of it so it will swell in the presence of certain measuring. So you can change, might swell in the presence of a certain pH. It'll swell by a factor of 8, up to a factor of 8. Swell pH or temperature. One thing we did, we took a hydrogel and we put one of our temperature sensitive ones and we put layers of graphene in it. Now graphene will if you shine an IR light on it, will heat up really efficiently. And so what we did was we got this guy to originally put in the middle these little magnetic alginate beads, which are biocompatible. This is from David Mooney's lab at Harvard. And it would then, as we released it, it would close up and hold those. We'd move it around and then we could shine IR light on it, would open and release those. And that's a cover of some journal recently, last year. That's one way we've been looking at doing it and controlling them with, in this case, IR light, but you could imagine if it got to a certain pH or certain temperature, it would do the same thing. Another one is we do have a way with magnetic fields to do it. It's a paper. We're filing an invention disclosure right now on the device, and it's a paper. I've got a draft. I just left the last draft back when I came. Hopefully it'll be published later this year sometime on how we can create devices that have relative motion on them with these same kind of fabrication procedures with a little bit of a different aspect. So we're going there.
You say that you'd like to have autonomy at an individual scale. Currently, it seems like you have one overall system, and each one of these little things is, I guess, a manipulator. At what point do you see it transitioning from central system, little manipulators, to each one being autonomous?
Yeah.
In terms of their capability, when does that transition?
Well, so one thing when I take my big electromagnetic system and I just give random inputs and I use so when it becomes like a bacteria, I consider bacteria fairly autonomous. Okay, the way a bacteria tracks something. So there's a food source over there, like one of the Marshall's thing. It's putting off some chemical. Bacteria are just randomly moving around. They're just they're tumbling. They're randomly moving. But as soon as they sense a molecule they like, they go a little bit further in that direction before tumbling. So it's this biased random walk thing. And eventually, then they'll find there's all sorts of interesting mathematical proofs about the way this works. Now, one thing that I think I would like to be able to do are create devices which are, maybe they're energized by random inputs, random magnetic fields, but they always feel like heading in a particular direction. Then I consider them autonomous, right? When I don't have to track them, when I don't have to use visual servoing to make them go there, when I can just give random inputs in it, and then they start heading along these gradients and ending up in the locations we want, then I would call them autonomous. So, you know. No, we haven't really done any. I mean, I've had people talk to us about it. We haven't done anything where we've carried stem cells. We do have a couple papers, a paper on magnetic kind of tissue scaffolds. This is with a group I work with in Korea, where we took a cage and we put cells in it that was magnetic, and we could move that cage around, and then it would sit there and the cells would kind of proliferate. And again, that was something on the cover of something. Advanced Materials. And that's about as close as we've gotten to, creating these kind of structures that can hold cells. You put them into location and then they go there. But I think one of the things I'd like to look at, it depends on who I've talked to different people and I get different answers, but I think there has been some efficacy shown, for instance, in stem cells injected into the heart, but I don't know how. I get different stories depending on who I talk to on that. Now, it seems like our catheter would be good at that, but then I talk to other people and there's like, well, there's already devices that do that on the market, but I don't know if that's true. You get a lot of opinions from doctors, and you have to take a vote and decide what that is. But yeah, I think there's certainly an area to go in, and also on the retina, we see things there, but we haven't gone that way. We talk to people now. So.
device too large, or?
Well, so, I mean, there are people working, you're talking about interventional. Right, yeah, I hear different things on that. People that have been in the field longer than me think, maybe Cam can correct me if, but you know, some people say, I've heard people say the only interventional MR that they think makes sense is the implantation of structures in the brain, deep brain stimulation, because you're targeting very specific areas. I don't know if that's true. I know as we go to, I can see why we're going to 7 Tesla and 9 Tesla systems, because we're going to get to the point where we do want to target particular cells in the brain for some of these diseases. But now, how are you going to do an interventional procedure and something like that? And these systems are going to be phenomenally expensive. And so then it also comes down to a calibration issue between all this. But with the fluoroscopy, also there's a lot of stuff you don't see in the fluoroscope, of course. And radiation. We do have a paper, I think it's going to be at ICRA maybe, where we took our fluoroscope and we've moved it around and done just some simple Kalman filtering, but just trying to get some 3D information. And I surprisingly we didn't find the literature on that. It was a pretty straightforward idea, but you know, CT does certain things, MR does certain things, but they don't do everything. And so the challenge is to find those problems where we can address, and I think it's just a case-by-case basis for us. At least we're still at the point of learning these things.
Any other questions? Well, let's thank again Brad for wonderful talk.
Thank you. Thank you.
Thank you so much.
Thanks for having me.